Dear audience, today I'm going to talk about bile accelerate carcinogenic processes in pancreatic ductal adenocarcinoma cells through the overexpression of MAC4. First of all authors have no conflict of interest. Pancreatic cancer is one of the leading causes of mortality rate globally and usually associated with obstructive jaundice. Our aim were to characterize the effects of bile acid on carcinogenic processes using a pancreatic ductal adenocarcinoma cell line and to investigate the underlying mechanism. First of all, we measure the concentration of bile acid in human serum obtained from different patients. After we treated kappa-1 cells with these serums, we found that first changes in the cell morphology, and the PDEC serum promotes the epitel-mesenchymal transition, and the PDEC serum with obstructive jaundice significantly increased the rate of proliferation, adhesion, and viability of the kappa-1 cells. In order to investigate the effects of bile acid in tumor progression, we treated kappa-1 cells with different bile acids for 24, 48, and 72 hours. After the treatment, we found that most of the bile acids significantly increased the rate of proliferation, adhesion, migration, and the ability of colony forming of the kappa-1 cells. So our first conclusion that bile acid accelerate the cancer processes in pancreatic ductal adenocarcinoma cells. So we wanted to know the underlying mechanism. We know that mucins are glycoproteins and have a significance in many cancer types. And we focused on MAC4, which is a biomarker in pancreatic cancer. And overexpression of this gene is associated with poor prognosis. Our question was, is there any relationship with other mucin expression and the tumorigenic potential of bile acid? We found that most of the bile acid time and dose dependently increase the expression of MAC4 in mRNA and in protein level also. We investigated these MAC4 isoforms in human samples, and we found that in PDEC patients with or without obstructive jaundice, we found a very strong MAC4 staining, but in the normal samples and the neuroendocrine tumor, the staining were completely negative. In order to clarify the role of MAC4 in carcinogenic processes, we regulated this MAC4 isoform via sgRNA transfection, and we found that MAC4 act as an oncogenic mucin. So our result shows that bile acid accelerate the carcinogenic processes in pancreatic ductal adenocarcinoma cells, in which the increased MAC4 expression plays an important role. So our data suggests that in PC patients where the disease is associated with obstructive jaundice, the early treatment of biliary obstruction improves life expectancy.